Growth factors are essential organic compounds that cannot be synthesized by an organism by itself, because they lack the genetic and metabolic mechanisms to synthesize them. Growth factors like essential amino acids, vitamins, purines, and pyrimidines must be provided as essential nutrients. In fact, bacteria can obtain these growth factors by adding organic materials such as yeast extract, corn steep liquor, liver infusion, malt extract, etc. into culture media. Here is a list of some growth factors and their corresponding function. Different growth factors are involved in different biological pathways. For example, folic acid is important for 1-carbon metabolism and methyltransferase. Vitamin B1 is significant in decarboxylation reactions. Vitamin K is critical for electron transfer. Mineral salt is another source of essential nutrients. It is divided into microelements and trace elements. Microelements include phosphorus, sulfur, potassium, magnesium, and calcium. For instance, phosphorus is the key component of nucleic acids and the phospholipids of a cell membrane, which serves in energy transfers. Bacteria can absorb phosphorus from inorganic phosphate ion as well. The other critical element is sulfur, and it is an essential component of sulfur-containing amino acids and vitamins such as thiamines and biotin. The important natural inorganic sources of bacteria are from sulfate ion and hydrogen sulfide. Macroelements such as potassium, magnesium, and calcium are often applied as cofactors for enzymes during biological activities. In case of insufficient mineral salt, these elements are sometimes added into a lab cultural media. Most trace elements are usually supposed to be cofactors and essential for the functions of certain enzymes. For example, copper is cofactors of cytochrome C oxidase. Iron is important for catalysis and peroxidase. Zinc is critical for carbonic anhydride, nucleic acid polymerases, and so on. Bacteria can utilize trace elements from tap water and other components of media. Water is an extremely important component of microorganisms. The percentage of water content in bacteria, yeast, and molds are 80%, 75%, and 85% respectively. Microbial spores contains relatively lower percentage of water content. The moisture content of the mold spores is approximately 39%, while there are less than 30% moisture content for endospores core. The main determinants of a microbe's nutritional type are its source of carbon and energy. We saw earlier that microbes can be defined according to their carbon as autotrophs or heterotrophs. Another system classifies them according to their energy source as phototrophs or chemotrophs. Microbes that photosynthesize are phototrophs, and those that gain energy from chemical compounds are chemotrophs. Autotrophs are bacteria using inorganic substances as their carbon source and energy source. When autotrophs use light as its energy source, it is called photoautotrophs. Depending on whether oxygen are made by the bacteria, photoautotrophs are divided into oxygenic photosynthesis and anoxygenic photosynthesis. There is a special group of bacteria that survive totally on inorganic substances. It's named as chemoautotrophs. For instance, methanogen, a kind of chemoautotroph, produces methane gas under anaerobic conditions. Heterotrophs are bacteria derive both carbon and energy source from organic compounds. The majority of bacteria are chemoheterotrophs, which relies on aerobic respiration. Aerobic respiration means bacteria convert biochemical energy from nutrients into adenosine triphosphate, ATP, the energy source. Chemoheterotrophic microorganisms belong to one of two main categories that differ in how they obtain their organic nutrients. Saprobes are free-living organisms that feed primarily on organic detritus from dead organisms. For example, some opportunistic pathogen and facultative parasites can feed on dead organisms. Parasites ordinarily derive nutrients from the cells or tissues of a host, for instance, some pathogens and obligate parasites. The main determinants of a microbe's nutritional type are its source of carbon and energy. We saw earlier that microbes can be defined according to their carbon as autotrophs or heterotrophs. Another system classifies them according to their energy source as phototrophs or chemotrophs. Chemotrophic microbes can utilize chemical energy from chemical compounds, both organic and inorganic substances. Autotrophs derive energy from one of two possible non-living sources, sunlight and chemical reactions involving simple chemicals. The majority of heterotrophic microorganisms are chemoheterotrophs that derive both carbon and energy from organic compounds. Processing these organic molecules by respiration or fermentation releases energy that is stored as ATP. An example of chemoheterotroph is aerobic respiration, the principal energy-yielding pathway in animals, most protozoa and fungi, and aerobic bacteria. Compared to common, familiar organisms, chemoautotrophs have adapted to the most stringent nutritional strategy on Earth. These prokaryotes survive totally on inorganic substances such as minerals. Photoautotrophs are photosynthetic, that is, they capture the energy of light rays and transform it into chemical energies that can be used in cell metabolism. Depending on whether oxygen are made by the bacteria, photoautotrophs are divided into oxygenic photosynthesis and anoxygenic photosynthesis. For example, algae and colorobium, respectively. Green non-sulfur bacteria or purple non-sulfur bacteria that derive carbon source and energy source from organic compounds and light are classified to be photoheterotrophs. This table lists all the nutritional categories of microbes by energy and carbon source we have learned about previously. Please remember that most microorganisms are chemoheterotrophs, includes many bacteria, protozoa, and fungi.